Hello everyone. Welcome to Enzyme MBBS, where complex medicine becomes simple and exam ready. Okay, without wasting any time, let's get started. So, writing the answer for the cellular events of acute inflammation begins with the introduction. So, give a point of introduction that is, acute inflammation is characterized by recruitment, activation, and actions of leukocytes, mainly neutrophils initially, followed by monocytes or macrophages. A straightforward sentence. Next, write about the leukocyte recruitment to site of inflammation. Now, what is this leukocyte recruitment? You know what is a recruitment, right? It is basically electing someone to a particular job. Similarly, now leukocyte is elected to perform something specific. Okay, that's why we call it as leukocyte recruitment. Now, where it has been sent, usually recruitment word is used in a police job, right? So they say like you have been recruited in this area and all that. So now this leukocyte has been recruited in the site of inflammation. It has to go to the site of inflammation and there it has to perform the specific job that is assigned to it. So now they have to see leukocytes are recruited from blood to extravascular tissues by a multi-step coordinated process mediated by adhesion molecules and cytokines. So that is what we are going to see in this video. So before explaining each step in that multi-step process, in order to show the examiner that you know the whole process, just write a sequence of events in an overview form. So basically, first vasodilation and stasis happens. This we discussed even in the before video, that is vascular events of inflammation. So basically, the histamine mediated vasodilation happens and the stasis takes place, right? And after that, what happens? Margination happens. Now again, some of you may have some confusions like what is this rolling, what is this adhesion, what is this transmigration. Everything we are going to cover in this video. So once you finish the video, then you can easily write this process, this step, but keep it in a beginning. Okay, so that is vasodilation and stasis, margination, rolling, adhesion, transmigration, and chemotaxis in tissues. Now, let's start with the margination. So, due to vasodilation, blood flow slows. That is stasis. We discussed it in the vascular events. And if you have not watched that video, I have given the link in the description. Go and watch it. You can understand it. Then RBCs move centrally, leukocytes displaced to the periphery. It's like, see, the blood flow. The velocity of the blood flow is slow, right? So because of that, the contents which are present in the blood, which was moving along with the current of blood, now they're gonna go to the peripheries. So this leukocyte has been displaced to the periphery. So in the periphery, what is present? Endothelial cells are present. So leukocytes come into contact with the endothelium, which is basically known as margination. The next process is rolling, which are mediated by the selectins. Now in the diagram, you just see that before inflammation, the selectin molecules are inside the endothelial cells, right? And once the inflammation takes place, these mediators, that is interleukin 1, 8 and tumor necrosis factor, you will learn more about this in one more video that is about cellular mediators of inflammation. So basically, they are some mediators which are gonna take part in the inflammation processes. So what they do, they activate the selectin molecules. The selectin, which was present inside the endothelial cells, now will come out. And there are some specific receptors in the neutrophils which are gonna bind to this selectin molecules which have been projected out. So that's what is happening in the second diagram. There is something called a Cialyl-Lewis X receptor in the neutrophil. It is gonna get bind with the selectin molecule, and since this selectin is present in the endothelial cells, it is known as E-selectin or endothelial selectin. So basically what happens is there is a weak bond between selectin and the ligand. Ligand is nothing but here Cialyl-Lewis X receptor. So it is a weak bond, something like van der Waals. So this is weak. So imagine a neutrophil is going to get bounded with the selectin molecule weakly. Now the blood flow detaches it. It pushes it. So it again, what happens? It detaches the bond. Then again, they're going to bind to the next selectin molecule. Again, the blood flow is going to make it detach. Again, it's going to bind to next. So it's a kind of a rolling process. I hope you can imagine. So that's why we call this process as rolling. And you have to write the selectins which are involved here. So that is basically E-selectin, P-selectin, and L-selectin. E is endothelial selectin, P is for platelet selectin, and L is for leukocyte selectin. So the ligands are also mentioned here. So for E-selectin, it is Cialyl-Lewis X. For P-selectin, it is PSGL1, and for L-selectin, it is Cialyl-Lewis X only, but it is now present in the endothelium. If you carefully observe in the diagram, the Cialyl-Lewis X receptor is present in the neutrophil, or I can broadly say WBC. But in the case of L-selectin, the ligand, that is Cialyl-Lewis, is present on the endothelium. So that is the difference. Okay. Now next, leukocyte activation happens. Now in the flowchart, you may have studied that after rolling, the adhesion comes. But before adhesion, these leukocytes have to be activated. Why there is a weak bond? Because the integrins they are present there are of low affinity type. Okay, so that low affinity has to be converted into high affinity so that they can bind, they can stick, so that the blood cannot detach it. Okay, so that thing happens here in the leukocyte activation. So there is something called as chemokines. Again, we're going to study about them in cellular mediators video. So these chemokines, they are produced at the site of infection. What they do, they get bind to the proteoglycans. Now you may have doubt, what is proteoglycans? So proteoglycans, if you have studied your cellular membranes in the first year very well, there you can you would have heard that on the cell membrane there will be proteoglycans, glycoproteins, or something like that. So I hope you can recall it. So this chemokines can go and bind to that proteoglycans on the endothelial surface, and what they do, they bind to the leukocyte receptor as soon as leukocyte comes, and they give something called as inside out signaling. And because of that, the integrins that are present in the leukocyte, which are of a low affinity type, gets converted to the high affinity type. So that's what basically we needed. So now they are the high affinity type of leukocytes. Now the adhesion happens, which is integrin mediated. So I have given in the boxes like which integrin is present in the which leukocytes and what are the ligands here. Basically that integrin is going to bind with the ligand. Okay, so this is completely a memorizing part, because there is no logic why it is called as LFA1 or why it is MAC1. So you just have to mug this up, because they are basically names, right? So that is what, but I hope the adhesion process is clear. The high affinity integrin is going to get bind with the ligand, and because of that, the now what happens, the leukocytes are adhered or they are arrested. Before that, what was happening, they are going to bind and they are going to roll because of the blood flow and because of the weak bond between selectin and the ligand. But now the integrin has been converted into high affinity, so they are going to bind tightly, so the arrest of leukocytes happens. Okay, then comes the transmigration. Transmigration is also called as diapedesis. So that occurs mainly in the post-capillary venules, in which basically leukocytes, the whole soul and what you say, yeah. So the whole purpose of this process is that we have to take the leukocyte to the site of injury from the blood vessel. So they have to come out of it, right? So that's what happens in the diapedesis. So the leukocytes pass between the endothelial cells. So there are some key molecules which are involved in this process. So that is one is in the leukocytes, they will use some enzyme called as non-specific collagenase. Now, why non-specific collagenase? And you may have a doubt, endothelial cells contract, there is some space between endothelial cells, they can just escape through that. Why enzymes are required and all those things? See, below the endothelial cells, there is something called as basement membrane. And that basement membrane is made up of type 4 collagen, right? So it's not that easy for them to escape. Now, if they use specific collagenase enzyme, then the whole basement membrane is going to get disrupted. So if it is going to get disrupted, then what happens? Then your cell functioning is also going to get affected. So they use non-specific collagenase, which makes some tiny holes, and through that they are going to get squeezed out, or they are going to get come out with the help of one more important key molecule called as PECAM1, which abbreviates like platelet endothelial cell adhesion molecule. So they are present in the both leukocyte and endothelial cells. So With the help of these things, the leukocytes enter the extravascular tissue. After this, you have to write about the chemotaxis. Now, what is chemotaxis? So, leukocytes have come out of the blood vessel, but it may not be necessary that near there only the pathogen or whatever the factor that is causing the inflammation is present. It could be a little far away. So, to that particular point, now they have to travel. How they are going to travel? So that is what nothing but chemotaxis. It is leukocytes migrate along a chemical gradient towards site of injury, and it is a unidirectional movement. Not like leukocyte will go there, they will perform the phagocytosis and come back, and they re-enter the blood vessel. If you remember in which video we discussed this, in one of the video we discussed that there is something called as physiological apoptosis, and in the physiological apoptosis we spoke that apoptosis of neutrophils after phagocytosis. So that's what happens with them. So it is a unidirectional movement. How this happens? This happens due to there is something called as G-actin and F-actin molecules. Nerve muscle physiology, of course, you would have studied in the first year. So in the diagram it is mentioned G-actin, then the polymerization happens, it gets into gets converted into F-actin, and because of this they will move like this. Like if you see in the diagram, there is a kind of a finger formation. Now again they will become ovular, then again a pseudopode. Like that they will travel on basis of a chemotaxic agent. A chemotaxic agent can be endogenous or exogenous. Under endogenous, you can mention chemokines, complement fragment C5a, and arachidonic acid metabolites. And under exogenous, you can mention bacterial products. See, one thing I can understand that you may feel like so many names. What is the logic behind this? The problem is the complement fragment or C5a. It comes under immunology. So arachidonic acid metabolites, chemokine, all these are cellular mediators. So there is a separate video will be produced on them. There we will talk about that. And exogenous bacterial products. Then phagocytosis. Now again, phagocytosis is a complete 5-mark question. You can ask only the phagocytosis. It's that long. So, but in the question of cellular events, you have to just mention just a phagocytosis here. Okay, so that's what we have done here. So first, what happens? Recognition and attachments. Now, this happens via opsonins. Now, what is opsonins? You can imagine there is a kid who is struggling to eat chapatis. Okay, his mom is trying, but he's very struggling to eat chapatis. He's saying that I'm not going to eat it, or it is very difficult for me to eat it. So what his mother does, she dips that chapati in the rajma and she gives him. Now he's loving it. The smell of that rajma itself is like a eat me signal for him, and he's gonna eat it. And he's gonna ask his mother more. Give me, I will eat it. So the eating become easy because of that aromatic rajma, which was a eat me signal for him. Similarly, this opsonins, they're gonna go and bind to the particular pathogen or whatever causing inflammation. Now, as soon as they bind to it, they send a eat me signal for the neutrophil. So now they're gonna come and eat it. It's that simple. So here I have mentioned some opsonins. They are IgG, C3b, and collectins. So they act like opsonin, and that helps in the recognition. And next, engulfment. So for that, there will be formation of phagosome. Now, phagosome formation takes place, and then it's gonna fuse with the lysosome to form phagolysosome, and that phagolysosome will perform and kill. How killing or degradation happens? It can be oxygen dependent or oxygen independent. So oxygen dependent, it is the respiratory burst, a reactive oxygen species via NADPH oxidase, myeloperoxidase to the HOCl, and hypoiodic acid. Now I can understand you can't get what is actually happening here. Oxygen dependent, oxygen independent, what is respiratory burst, what is the reactive oxygen species via NADPH oxidase. That's why I said we're gonna discuss this whole phagocytosis process in another video. In this answer, you have to mention this. I will suggest you that the whole chapter, if you're gonna make playlist, we are gonna put all the answers. So when you are studying for the final exams or whatever exams or as you study, watch that whole playlist so that you can get the complete idea of that chapter. Once you get that, then when you're gonna write some answer in the exams, this is how you have to write. So that's why I've mentioned in answer format. For time being, you just remember what all to write in this particular question. The explanation part of this is going to come in another video. Okay, so oxygen independent, it is lysosomal enzymes and defectants and lactoferrin. And last but not the least, you have to write about the leukocyte type and temporal pattern. Now, I know all of you know that in the acute inflammation, neutrophil is the predominant cell. And if neutrophil fails, then macrophages comes into the picture and slowly it turns into chronic. My question is, why neutrophil? Why not macrophages? Or why not eosinophils? Why it has to be neutrophil in acute inflammation? So the reason for the neutrophil predominance is, one is it is most numerous or more numerous. You would have studied in your first year physiology, right? The neutrophils are the most abundant type of WBC. So that is one reason. And one more is they respond rapidly to the chemokines. Now again, chemokines, we heard a quite few times this name in this video. So they are very much important. So for them, they respond rapidly. That's why neutrophils are more, what you say, that neutrophils are predominant. And one more is it has a short lifespan. So it is easier to perform apoptosis, physiological apoptosis, removal of neutrophils after the phagocytosis, right? So for all these reasons, neutrophil is the predominant type. And just write one more point for that extra mark, that is clinical correlation. So defect in any adhesion molecules, it will result in leukocyte adhesion disorders. Examples are LAD1 and LAD2.